You can get definitions for these classes of enzymes from your textbook or many places online, probably in explanations and examples for your online homework. I'm going to summarize them here and then take some time to see how all the things you've been learning about understanding organic molecules come into play in biochemistry. So oxidoreductases, simple version, you add or you take away oxygen, bonds to oxygen, or bonds to hydrogen. Transferases, move a group from one molecule to a second molecule. I shouldn't abbreviate it like mole. Hydrolase. Hydro is water. Lase is cut. You cut it using water. A lyase is you cut it without water. Isomerase, you change which isomer it is for that molecule. A ligase is tying things together or binding things together. So okay, so you should copy this down or have it somewhere so that we can switch to looking at actual examples in biochemistry. What we're looking at here is part of the glycolysis pathway. It's a primary energy pathway in cells where you take glucose and you break it down eventually into a molecule called pyruvate. So you've seen glucose molecules before when you learned about sugars. And we're just going to follow through and see how we can identify four of the different enzyme classes in this pathway. So there's glucose. We look over here at this glucose 6-phosphate molecule. And there's one thing that's obviously different. This PO3 with a -2 charge replacing the H there. And what we've done is we've transferred a phosphate group, which is actually the PO4, replacing this hydroxyl group. And we transferred the phosphate group from ATP, adenosine triphosphate, to adenosine diphosphate. So it went from tri to di and gave that third phosphate group to the glucose molecule. So this hexokinase is a transferase. Let's go to the next one. Well, the name gives it away here, but if you look at these, the phosphate's the same, lots of carbon and oxygen and hydrogen, and if you go through and count, you'll find that it's C6, H11O6, and the same here, C6H11O6, and the PO3 2-, PO3 2-. So you can take the time to count those. This is an isomerase, the same number of atoms, but they're in a different arrangement. Let's move on to this one. If you check through carefully, everything's the same, except for here, the OH is replaced with O, PO3 2-. And triphosphate becomes diphosphate, so it's another transferase. These kinases are transferases. All right, we've got a new thing. Fructose, so that's our sugar fructose. 1,6-biphosphate. That means there are 2 biphosphate groups, 1, 2, and they are connected at carbons 1 and 6. Carbon, let's see, this would be carbon 1 because here is the hemiacetal. On this side, it's just, well, no extra oxygen here. So this is oxygen is part of the hemiacetal group. So that makes this carbon 2 and carbon 1. So that's 1. And then we count around 2, 3, 4, 5, 6. So lots of things that you know help you understand these names. Let's do the same with these names here. Glyceraldehyde 3-phosphate. In the fatty acids chapter, you learned about glycerol, which was 3 carbons, 1, 2, 3, with 3 hydroxyl groups. There's 1 hydroxyl group. This here was a hydroxyl group until it got replaced with a phosphate. So there is the 3-phosphate replacing one of the hydroxyl groups on glycerol. On the other hand, there's a carbonyl. This is an aldehyde group. So here the aldehyde has replaced a hydroxyl of the glycerol. So it's glyceraldehyde. That's carbon 1, 2, 3, phosphate. Now you can do the same here, but you might not have memorized what acetone is. So the one tells us it's a ketone. And here are 3 carbons, and at the middle carbon is a double bond O. Go to there, see that it's carbon on each side. That makes it a ketone. Now the 3-carbon ketone is propanone, but it has a name, also an official name, called acetone. So we've got acetone here if these 3 carbons and this carbonyl were all that we had. It's called dihydroxyacetone. So we're looking for 2 hydroxy groups. If we go to this carbon here, there's a hydroxy group. Go to this carbon here, there's almost a hydroxy group. It's a phosphate group. This would be the second hydroxy, except it's phosphate, dihydroxyacetone phosphate. So we take one of the hydroxy groups, replace it with a phosphate, and that's where the name comes from. So you know all the pieces of these names. You couldn't maybe write them down, but you can understand them if you take a pause and look at them critically and say, okay, I have learned this stuff before. Let's go back to the reaction. Fructose 1,6-bisphosphate. If you count all of the atoms, the formula is C6, H10, O6, plus 2PO3, 2 minuses. Let's go down here. You can write that down. C6, H10, O6, and 2PO3 minus 2 minuses. Down here we've got C3, H1, 2, 3, 4, 5, O3, and 1 PO3, 2 minus. If you go to the other side, it's the same. C3, H5, O3, PO3, 2 minus. Add those up, they equal the C6, H10, O6, and 2 PO3, 2 minuses that we started with. So we've split this molecule in 2 without adding any water. We've cut it with no water. That makes it a lyase. This aldolase is a lyase. Now here, dihydroxyacetone phosphate becomes glyceraldehyde 3-phosphate. We already counted the atoms. The connections are different. The double bond O is moved. This hydroxyl group has moved. This is an isomerase, as it's called there. Let's move over one. There's some clues. Glyceraldehyde phosphate dehydrogenase. Okay, let's take a look at it. This has 3 carbons, as we see, 3 oxygen. This now has 4 oxygen, 5 hydrogen, only 4 hydrogen, 1 phosphate, 2 phosphates. So we have 2 things going on here. One is the transferase. We go from having 1 phosphate and a double bond O here to having, well, the double bond O is still there, having an H here to having an OPO3 2-. So we're transferring a molecule. At the same time, we are removing a hydrogen there. So this molecule, and adding a bond to oxygen. So this molecule is both an oxidoreductase, dehydrogenase, D taking off a hydrogen, and a transferase where we're adding, oh, I'm sorry, we're not transferring a PO4, we are adding a PO4 from phosphate. This is could be called a ligase. It is an oxidoreductase primarily. Could possibly also be classified as a ligase. So the ability to classify these is not the most important thing you can do in the world, but what we're doing as we classify is we are seeing how all the things we learned in organic chemistry help us examine this more complicated biochemical pathway and understand what's going on. So going more quickly, this time it's ADP becoming ATP, so it's gaining a phosphate, and we go from 2 phosphates to 1 phosphate. This is a transferase. Mutase, interesting name, suggests mutation. If you count the atoms, you can pause the video and count them, you will find, well, here they've changed the charge from minus to OH, but we can really ignore that. That's a pH dependent thing. That's an acid-base reaction, happens really easily in cells, and we generally don't even consider acid-base reactions. We just assume, oh yeah, that can happen, happens in water all the time, and we ignore that part of the molecule. So here we've got an OH and a phosphate, and then on this molecule we've got a phosphate and an OH. So it's switched places. This is an isomerase. This mutase is an isomerase. And you can see in the name, the only thing that's changed in the name is it goes from 3 phosphoglycerate to 2 phosphoglycerate. So the name's even the same, telling us this looks like the same molecule, but where we attach the phosphate has changed. Oh, glycerate. 8 should be familiar from carboxylates. Here, it shows it as a carboxylate, so glycerate. Here, it would probably more accurately be called glyceroic acid. But again, because these H pluses can go on and off so easily, we often just call it by one name and don't bother to switch the name, even if the charge state shifts. Let's look at this next one. What's different? The 3 carbons are the same. The carboxylic acid is the same. The phosphate is the same. What's different is That hydrogen's gone, and the OH is gone, and there's a double bond. So we've taken off water. That's shown with this arrow going out. This arrow goes backwards. If we're going backwards, we've got water going in, shown with this part of the curve. So we're going this way, and we're taking water out. This is, we've changed the number of bonds to hydrogen and to oxygen. So this enolase is a kind of oxidoreductase. And here we've got another kinase. We've seen kinases before, transferring a phosphate group. This is a transferase. We have seen most of the types. We're going to look at the citric acid cycle. See another ligase. Here, if the citric acid cycle needs this molecule oxaloacetate, shown here, 1, 2, 3, 4 carbons, and carboxylic acid, a ketone group here. It needs this in order for it to make citrate and go around the cycle. So if for some reason your oxaloacetate is low in the cell, you need to make more of it. One way to make more of it is to take pyruvate. That's this molecule here with 3 carbons, 1 carboxylic acid, a ketone, and go around this way. Let's follow it around to oxaloacetate. And what does it require? It requires the enzyme pyruvate carboxylase. And here is a carbonic acid molecule and energy. The ATP doesn't actually get used in making our new molecule, it just gets broken down to give energy. So here's the 4th carbon added on. So we go, here's pyruvate with a carboxylate and a ketone. Here we have a carboxylate and a ketone. There's a carbon with 3 hydrogen. Here's a carbon with 2 hydrogen and an HCO2 added. Here's where the HCO2 comes from. And then there's an extra oxygen that it doesn't show us where that goes. Would need to understand more about the mechanism of the reaction. But we've added on a 4th carbon to this 3 carbon molecule here. That is a ligase. If you follow this reaction around, you will see a lot of reactions where the number of bonds to oxygen and hydrogen changes. We lose an oxygen there. Here's a single bond. The oxygen's gone and a double bond is formed. Here we lose some hydrogen atoms. Actually, and a CO2 comes off. This is another, it's called a dehydrogenase. This is another oxidoreductase, Got a transferase here, transferring this part of the molecule. Sorry, not a transferase. We did 2 things here where we took off a CO2 and we attached it to CoA. So we've got an oxidoreductase and a transferase reaction going on here, going on here, we are breaking this bond and that gives off quite a bit of energy. We're putting in an O instead. So this goes from a thioester to a carboxylic acid. I'm not actually sure how to categorize that offhand. I may add a note to the video. Here we've got a single bond becoming a double bond. So we've lost 2 hydrogen. That is an oxidoreductase. Here we're adding water in across the double bond. Again, an oxidoreductase. Another oxidoreductase, where we go from a single bond to a double bond to the O there. And then, as we go from this, 4 carbons to 5 carbons, this synthase is a ligase. So this synthetase is probably also a ligase, but I'll have to double check that. So we've seen ligases, we've seen oxidoreductases, we've seen transferases, isomerases, we've seen everything except for a hydrolase. They happen to be the most plentiful kind of enzyme, the most diverse kind of enzyme, but we haven't seen any of them in these pathways we've looked at. One place you find it is in proteases that break down proteins. So we're learning about proteins this chapter. Here is the peptide bond, this amide bond. And when we add water and move electrons around like these arrows say, it splits apart into an amine and a carboxylic acid. So we've added water to cut it. That makes it an example of a hydrolase. And there are numerous proteases that function in different parts of the body and different parts of the cell. So now we've seen in biochemistry, it's a little bit painful going through and counting atoms, but we've seen all the different types, the 6 major classes of enzymes, and seen how all the things you learned in organic chemistry can help you identify these just by looking closely at the changes in atoms in these molecules.